When an HLA gene encoded in the DNA of chromosome 6p is transcribed, a ribosome binds to it and inserts enough amino acids into the sequence to expose a signal, which will then bind to a signal recognition particle. This translocates the ribosome with the RNA attached to the endoplasmic reticulum, where the remainder of translation will occur in the lumen of the endoplasmic reticulum. It is here that the HLA protein will bind the beta 2 microglobulin, and then also where it will bind a peptide from the breakdown of a protein inside the cytoplasm. In the cytoplasm, a complex known as the proteasome digests proteins, both proteins from the cell and then potentially those from abnormal cancerous cells or from virally infected cells to produce peptides of 7 to 11 amino acids. The peptides digested by the proteasome then pass through an ABC cassette transporter known as TAP, the transporter associated with antigen processing, which is more complex than is depicted here. It forms part of a multi-protein complex where the HLA protein, the beta microglobulin protein are together. It was depicted in a simpler fashion in this animation. Because of this transport, then peptides from the cytoplasm, whether they be from proteins in the cell or perhaps viral proteins, are then added to the MHC molecule. The MHC proteins then are transported throughout the endoplasmic reticulum and sent via vesicle to the Golgi, and from the Golgi then are shipped to the cell membrane, where they become cell membrane proteins presenting a peptide from the interior of the cell on the exterior, where it can interact with cytotoxic T cells.